Hi everyone! I am Saniya Kule and this little cutie is cute little Yangshi. Woo! Saying hi, hi. And in this video, we'll be talking about generating a sequence logo from a DNA motif model. So Yangshi, do you have any logos besides being cute? So let's get started. A biological motif is basically a pattern that's found to occur in a set of biological sequences like DNA sequences, RNA sequences, protein sequences. It can be like an exact DNA sequence, for instance, or it can be a degenerate, there's no go yang, she says, consensus sequence that you can use ambiguous characters like R can represent adenine or guanine. You can also describe biological motifs by a probabilistic model like position-specific scoring matrix, PSSM, or a probability weighted matrix, PWM or P matrix. And I talk about this a bit in these videos. I talk about a probability weighted matrix where, for instance, you also, we include the background as well, but really this is the part of the motif. And for instance, this motif would have these 9 bases here. It's 9 units wide, and here ACGT, and each column represents the probability of having that given base in that particular position of the motif. And here we're kind of like selecting the highest values here. And if there's not one, if it's not too high, like I think the threshold in this image is like 0.8 or something. So if one base has a 0.8 and above, it's colored in red. Otherwise, it's in like light blue here. And the idea is that, for instance, we have a 97% chance of an A in the first position of the motif. So all of these sequence logos, there are 9 spots, each corresponding to this here. So we talk a lot about the probability weighted matrix, and I have another video just dedicated to calculating the height from such a matrix. Probability weighted matrix directly to the sequence logo. But in general, a sequence logo in bioinformatics is a graph representation of the sequence conservation of nucleotides, the RNA or the DNA, or amino acids like protein sequences. So we typically build a sequence logo from aligned sequences. Or we can also get them from a probability weighted matrix, calculating the information content there. But I do that in another video. In this video, we're going to look at it from aligned sequences. But in general, the sequence logo shows the consensus sequence and the diversity of the sequences. So from a sequence logo, we can learn like sequence characteristics like protein binding sites in DNA or functional units in proteins. So for instance, the higher the height is, the more information we have. Like we can say that in this region, it's high chance that this is an A, a G, a T, T, C, for instance. It can kind of guide that for us. Y-axis is in bits here. The sequence motifs, I cover again in another video going from probability weighted matrix to this sequence logo, but we can similarly go from this set of aligned sequences, and this is what we're doing in this video. Our focus is on this here. We can have a set of aligned sequences bound by a transcription factor. Then I show you here that in our video, we kind of think of these as like motif occurrences. So our model, whether it's Gibbs sampling or NEEM for motif discovery, we are finding motif occurrences out of a long list, not a long list, a long set of DNA sequences. We've identified where we think the motifs are occurring, and so we had a lot of DNA sequences, kind of long, and we were told that that was kind of where the transcription factor was showing some strong activity. And from that, we're trying to infer where those motifs could be, like where is that transcription factor potentially binding. And when we identify those regions, we just sort of like put them together. We say, okay, these are the motifs of a given width. Like here, this motif width is 6. 1, 2, 3, 4, 5, 6. And I like to include in my probability weighted matrix the background, which is a non-motif part. But in general, the position weight matrix or probability weighted matrix would be for the motif parts. So we put the motif occurrences here of the same width, and they are aligned. And we say that we believe that these could be bound by the transcription factor. And this part here is kind of like what we can also infer from the position weight matrix. And I do show like how we can kind of go from one to the other and vice versa in my other videos, so please do see those. But here we're going to see how we can go from a set of aligned sequences bound by a transcription factor, for instance, and then infer this probability weighted matrix logo, position weight matrix logo, or sequence logo. So in general, we align the related DNA, RNA, or protein sequences or DNA sequences. They have common conserved binding sites. So the most conserved parts can create good alignment. So if we notice something that is pretty much there a lot, then that is great. For instance, what we can notice here is that these sequences seem to have like G and A, and like this part, these sequences seem to conserve this. So when we check out these corresponding positions in the sequence logo, that is 1, 2, 3, 4, 5, 6, we notice that the heights of G and A are really high here. And also, I think I did not see this, T and A too. They have this GATA, and yes, my bad. So these four are conserved, GATA, GATA, GATA. And then we see this over here, the sequence logo is kind of showing us that, hey, you have the maximum information 2 bits here for GATA. So, in DNA, I just want you to see that the residues, when we say residues, those would be DNA bases, ACGT. For proteins, those would be like amino acids. We use the position weight matrix. I have another video, again, so Yankee saying please see that, about how you calculate those heights and the information content from a position weight matrix to construct this. There's another video, please see the link in the description. But here, we can look, if we align these sequences that are bound by their transcription factor, we then are using multiple sequence alignment to create the sequence logo from the set of sequences. Otherwise, again, if we're using the position weight matrix to make this, then I have another video on that. But this sequence logo or PWM logo, it shows how well these residues, these DNA bases, are conserved at each position. The higher the number of residues, the higher the letter will be, because the better the conservation is in that position. So all they're saying is that we look in a particular position, like, we look in position 1, and then we count how many times do we see a particular base. Well, we see 2 G's and 2 C's here. It's kind of in the middle, but here in the second position, we see all of them are G's. So that's like 4 G's, so it's super high. 4 A's, 4 T's, 4 A's, super high. And then we see just 2 A's and 2 T's, so it's not as high. But when we had perfect, like 100% of each base in these positions, and this is super high because we have more information, there's more conservation of a given base. That's what it's kind of trying to show us. So, in a given position, we also do scale the different bases like T and A based on their frequency. So T and A each appear twice. So we divide it in half, so to speak. And the height here is a number of bits. These are a stack of residues of DNA bases. And this information is measured in bits, which is information content. So again, we can use these sequence logos to represent conserved DNA binding sites, like sites where transcription factors can bind, like TF binding sites, for instance. And from a probability weighted matrix, where for each position you have the probability of a particular DNA base, we can construct these here. But the same information is essentially represented using the sequence logo where the y-axis is in bits. But I just wanted you to see that we can also represent the same information from a probability weighted matrix in this fashion, where they all sum up to 100% and we're just seeing the relative breakdown. But it's like we instead are also calculating the height and then stacking based on like how confident we are of a particular DNA base in a particular position, which is bits. So instead of probability on the y-axis, we are going to use bits in our situation, which can be between 0 and 2. Using one of the most basic examples that we've seen. It's actually a very famous consensus sequence. It's a Kozak consensus sequence. You have a messenger RNA that has A, U, G, and C. But now when we start translation in the ribosome, like cute little organelles, that help translate the messenger RNA to a polypeptide chain, what we are noticing is that starting codon is AUG. This is the Kozak consensus sequence. And this AUG codes for methionine here, and then we go ahead and read every 3 DNA bases, and then we stop here. There are 3 stop codons. But AUG is the only universal start codon. And what you can see here is when people analyzed in humans the translation bases, they found this maximum height like at AUG here, and it was always at AUG. Then what they also did from the sequence logos is they started kind of looking at which ones also kind of occur, like A happens kind of on the top. We look at the top kind of the top is usually either the most frequent, has the highest probability of occurring that base, or it's tied with the next one. So we typically just stick with the top. So A, C, C, A, C, C. And then this one, we just choose G because it's on the top. So by and large, the sequence around the start is like A, C, C, A, U, G, G. But it's like this one is uniformly the start of all polypeptide chains. It's always AUG. This is the Kozak consensus sequence. ACC and G after. These are the bases that tend to be the most conserved around the initiation codon for all human messenger RNAs. If you look around here, there's not as much information here or here. So we really are just sticking with this here. I mean, this one is the same height as this, but the only reason is we just kind of want to say ACC because we just want it to be continuous. A, we think it's C and then C. But this is high, this is high, this is great, and then this is great here too. This is just showing you an example of how we use consensus sequences, how we use sequence logos for intuition.
To learn how you can take motif occurrences, whether, by meme, expectation, maximization, implementation, Gibbs sampling approach, or any approach that basically gives you motif occurrences, and how you can construct a sequence logo. I kind of reviewed some of the concepts in meme and Gibbs sampling, and I went through and explained how to interpret the output of the sequence logo in comparison with the probability weighted matrix. The PWM or the P matrix, the model, the motif model that we get. Yankee is saying that if you find these videos helpful, please like, subscribe, and share. And please reach out with any and all questions that you have. I hope that this is a helpful video for you guys. As Yankee saying, let's get started. Okay. We're going to be talking on generating a sequence logo from a DNA motif model. SpongeBob and Patrick, right? They're looking on the web and basically they're looking at how to create a sequence logo from a set of motif occurrences here. So for instance, we are going to be talking about the same example that I talked about in the expectation maximization for motif elicitation, the meme algorithm. It kind of sounds, just like a meme, and just like a meme has a sequence, like a meme has a sequence of words on top of it. Similarly, we're kind of going to be generating our little logo here, sequence logo, for what we generate from the expectation maximization and a few other examples, and I'll be motivating it. But the key idea is that, if we had a set of 4 DNA sequences I talked about in earlier videos, and please do see the links in the bio and my channel for more information that we talked about, how from these 4 DNA sequences here, each of 6 bases long, we discovered these 4 motifs here. And we're going to be seeing how we can basically input, CAG, GAG, GAG, and CAG, these motif occurrences, into this WebLogo application, and then generate a sequence logo that can kind of say that in the first position of the motif, it has a high chance of being either a C or a G. We don't see any other bases here. Then the second one always happens to be an A, so that's like pretty high up, and like pretty much 100% guarantee. And then G in third position, because the last one is always a G here. But we're going to talk about that and motivate that as well. So basically, what we're going to talk about here is on generating a sequence logo for a DNA motif model based on input DNA subsequences, sequences of motif occurrences in the original sequences. So in these original sequences, what are the motifs that we identified, like CAG, GAG, GAG, and CAG? So that's a preview. We'll be seeing how to generate the sequence logo. That's what Yangshi and I will be teaching you today. We'll be going through some examples and then some motivation biologically. As a little refresher, what I was talking about, in my earlier video. So please do see those. Please subscribe as well and check those out. But we had talked about a set of these 4 DNA sequences that are L equals 6 bases long. So they need to be the same length. So 1, 2, 3, 4, 5, 6, 6 of them. Each of, these are the 4 DNA sequences here. I just represented them in this nice little visual with my logo. My trademark logo is this star. That's my logo. Now, what's the logo of this transcription factor binding site, huh? Let's see. My logo is, and what you'll see here is that this is a successful star. But basically, these are the 4 sequences, each 6 bases long, and we chose this parameter. We could choose any parameter between 1 and 6 for the motif length. If we chose 1, it would just be 1 DNA base. So not really useful per se. If we chose 6, 1 DNA base could be useful for a single nucleotide polymorphisms, but for motifs, I don't think is really useful here. But 1 is not really useful. 6 would be like everything, which could also technically be the case. But in this case, we chose the motif width of 3 bases here. So we wanted to identify the 3 most likely bases, consecutive bases, the motif occurrence in each of these sequences that is most likely. And that could be like a transcription factor binding site or another key site of interest as well. So we ran through mean multiple expectation maximizations for motif elicitation. That's what mean means here. And we just tried to identify the best motif, for this set of 4 DNA sequences, each 6 bases long. And then this figure here is from this paper, how does DNA sequence motif discovery work as well, published in 2006. So how can we computationally extract an unknown motif from a set of target sequences? What are the principles behind the major motif discovery algorithms? Which of these should we use, and how do we know that we found a real motif? So this is an interesting paper here. But basically what they talk about in this expectation maximization algorithm, like mean, what you do is you start from a single site here in the target sequence that you believe could be the motif. So AAT, let me zoom in a bit. So AAT, CAGTT, and that's what this is representing here. So in fact, this part here is represented by this. So what we do is we start from the single site that we think could be the motif, and then we alternate. So we assign this site here in the target sequence to being the motif here. And then what we do is we update our motif model. So based on our selection of this, we say that our motif would then be AAT, CA, BCA, GTT, and the other bases are kind of squashed below. And then what we do is we will update our motif model. Then next what we'll do is we'll assign sites to a motif. Again, we update the motif model. Then again, we reassign the sites to the motif. And then again, we update the motif model, and so on and so on. So you can see, like, for instance, we are kind of moving towards like AA, something else. We are constantly updating our motif model till we identify the best model as well. So that's kind of how it goes, start from a single site and then we alternate between. And that's sort of what we did there. And I talk about this in more detail in my other videos. But basically, the expectation maximization mean multiple expectation maximizations for motif elicitation. It learned a probability weighted matrix model, the P matrix, which is basically telling us, that the optimal motif that I talked about was CAG. We selected it again because it had the highest here as well. But the spinal probability weighted matrix basically has a background, which is the non-motif positions here. And it also has the motif positions. This part is for the motif, and this part is for the background. And these are the probabilities of observing each base in the background in a non-motif position. 20.3% chance of an A in the background, 23.1% chance of a C in the background, 32.6% chance of a G in the background, and 24% chance of a T in the background. And then what we see here is the following. We see that for each position in the motif that we wanted to be 3 long, our width W is equal to, our width W was 3. We basically say that these are the probabilities that we would have a given base in a particular position of the motif. In the first position of the motif, we have a 16.1% chance of an A being in the first position, 37.3% chance of C being in the first position, 33.7% chance of G in the first position, and 12.9% chance of T in the first position, and so on. So in this one, in the second position of the motif, looking here at the second position, we have a 53.3% chance of an A being in the second position of the motif. So it's the highest for an A. And lastly, we had the highest probability for the G out of all of these. These all have to sum up to 1, as we saw. For all of these here, 100%, 100%, 100%, 100%. And the highest probability was for G being in the last position of the motif. It was 58.4% probability of a G being in the third and last position of the motif. The second highest was for A, so 15% chance of an A being in the last position of the motif. So kind of based on these probabilities, we were able to find those motifs as needed. So CAG was highest here. But the thing is that in sequences, okay, so the thing is that CAG is the highest motif because of the C here, the A here, and the G here having the highest probability. So what our approach would do is it would scan through this expectation maximization, will scan through these sequences. And its first priority is to look for CAG. If it identifies CAG, it's like, if it finds that part, the CAG, it's going to say, yep, this is where the motif is. That's the game plan. It scans along and it can find CAG here and here. But in other sequences, so CAG was here, this was the optimal CAG, CAG, and CAG. So these two were able to work out. This one worked out and this one worked out. What about the cases where there's no CAG in the sequence? What do we do then? We have to then look at the next likely motif. This seems kind of likely, this seems kind of likely, but CAG was 37.3%, right? So this is the optimal one. But if there's no CAG, we then kind of look in the direction. Can we see about the GAG? G has the next highest probability. So given A is there and then G is there, the given A is there and G is there, can we then look for GAG? Because we chose CAG because in this column, C had the highest probability. The first position. And we chose A because it had the highest probability in this second column. And then we chose G because it had the highest probability in this column. But if we don't have a CAG, like we don't have a CAG here, we don't have a CAG here. We don't even have a C in the sequence. So one of the reasons why we've chosen CAG here is C had the highest probability in the first column. Then A had the highest probability in the second column. And then G had the highest probability in the third column. But if we don't have CAG, this is no C at all in the second sequence. So how can we even work with CAG? There's no C here. And in the third sequence, there is CGG, but we could look at CGG, but it's not that optimal here either. So what we do next is we kind of find and we look and we see, oh, it turns out we can see a GAG here and a GAG here in the second and third sequences. They may not be the optimal CAG, but in fact, they're like the runner-up, because if we look at GAG, GAG would need, basically what GAG would do is it would stitch together this, and this. If we have a GAG, it's G, A, and G, and this is so close to this 37.3%. It's a runner-up, so this is GAG. That's why we select GAG here. It appears also in this sequence and in this sequence. So we kind of pass it in some ways. Let me put a little star here because we trooped through this. But that's sort of why we select these optimal motifs the way that we do. And then for the background probabilities, kind of based on what we observe, we can see that there's G here, G here, G, G. A lot of the sequences tend to have G in the background more. The background are these non-motif positions. So basically, if we were to cross out the motifs, the background is everything else. Majority are G, but there are other bases, as well. Like there's a T here and a T here and a T here. There is a distribution, but G is kind of prevalent in the background. So I just wanted to motivate that. So this probability weighted matrix is a final motif model that we learned. The probabilities of having each DNA nucleotide A, adenine, cytosine, guanine, or thymine, a particular position of our motif. So here K can be 1, 2, or 3 because our motif width is 3. We're learning 3 bases. So again, what we were trying to do is learn these 3 bases, Yangshi. That's what we were trying to do here. That was the whole point of this. We can then see that what we found earlier was the corresponding 3 base subsequences for each of these 4 sequences based on our Z matrix. So we had found, this position 1, 1, 1, 1. This means that this is where we should start. So we should start in the 3rd position for the first sequence. That's where the motif occurs, starting in the 3rd position. And for the second sequence, the motif starts in the 1st position. For the 3rd sequence, the motif that we identified starts in the 4th position. And for the 4th and last sequence, the motif starts in the 2nd position. So this part here, this is what this is capturing. This is capturing the columns where there's 1, and only in a row, it can only be 1 entry that is 1. So basically, again, you see this 3, 1, 4, 2 here. Eeks. So yeah, you see this 3, 1, 4, 2. This is what this means here. That in the first sequence, the motif starts in position 3 of sequence 1. So 1, 2, and 3. It starts in position 3 here. That's why this was a 3. It starts in the second sequence, it starts in position 1 at the very beginning. So that's why this is a 1 and a 1. And the 4th, it's GAG, which starts in the 4th and last position. 1, 2, 3, 4. Yep, it starts here. That's why this is a 1 and this is a 4 here. So motif is in position 4 of sequence 3. This is like the sequence number, sequence 1, 2, 3, 4, and this is like the starting position here. So yeah, this left is the sequence number. And lastly here, 1, 2, the motif occurs in position 2 of this 4th and last sequence. So motif is in position 2 of sequence 4. So we take these J values, and that's what these are: 3, 1, 4, 2, 3, 1, 4, 2. And that's how we interpret this. So based on this final motif model, based on our final motif model, we learned the subsequences in the original DNA sequences. And these represent where the DNA motif is likeliest to occur in these original DNA sequences. So in the first sequence, we identified CAG here, CAG here. In the second, we identified the runner-up sequence, GAG. And the third, it was also GAG, because after CAG, it's like the next most optimal, because G is 33.7%, which is a little bit behind the optimal of 37.3%. So we went with GAG. It was also found in both of these sequences, which, did not have CAG. So this is also why we selected it, like the multiple expectation model learned that. And so that's why we found GAG here for sequence 2 and GAG for sequence 3. And lastly, CAG for the fourth and last sequence.
Those were the sequences.
We had this set of 4 sequences, each 6 DNA bases long, and we chose a motif width of 3. So we could have chosen anything from 1 to 6, but you have to really be kind of mindful while you're selecting a certain motif width. And what we ended up learning, as I showed, is we ended up learning the starting positions of the motifs in these sequences here. So 3, 1, 4, 2. So what this means is 3 means is that we're starting. So these are the positions here, 1, 2, 3, 4, 5, 6, of these bases. So 3 means we started again in position 3. Then the 1 means again, we started in the first position here for sequence 2. Then this 4 means that we started in the 4th position of sequence 3, the sequence number. And then this 2 means that we started in the 2nd position of the sequence 4 here, CAG, 2nd position. And in doing so, we learned these subsequences. We learned CAG, we learned GAG, GAG, and CAG. That's what we identified from here. As we showed here, these were the subsequences. These are the occurrences of the motif model. So the key question then is, how do we find the sequence logo for this motif? So these are the occurrences in the original input sequences. We found CAG, GAG, GAG, and CAG. So how can we learn the sequence logo from this? This is representing what we learned in our probability weighted matrix. We applied it and also found what this would be here. So this is the question that hopefully I tied in multiple expectation maximization approach from motif elicitation. But now we're going to see, how do we get this sequence logo, Yangshi? How do we find this? That's the key question. Now we're going to see how to construct the sequence logo. And we're going to see how to construct a sequence logo for this motif given by these subsequences using this WebLogo application.
This is the WebLogo application that we'll be going to.
So we're going to be going here to this link, and we're going to import our learn motif occurrences here of length 3 bases. That's what we wanted. So we're going to go here. So I want us to output a PDF, like some higher quality PDF or PNG, and the logo size should be large, and the color scheme should just be classic. So we came here. We can also upload a file. Since this is relatively smaller sequence, I'm just going to put in the 3 base motif and these 4 occurrences. So basically, I'm putting in these subsequences as input. This is our input to the sequence logo motif, GAG, because this is what we have learned from our model. And so we put in these 4. So we input these 4 in here. Okay, so we input those 4 occurrences. Saniya's example with a little smiley. And then we can make it like a, a PDF or something like higher resolution. This is how we'd make it a PDF, for instance. And logo size, let's make it large. Color scheme is the auto, but there are other types that you could look at. So let's create the web logo. Let's okay, so actually maybe just don't put a title in right now. I could mess things up. So we can just create the web logo here. And what we observe is that this is indeed a very high quality sequence logo. Yangshi, this is the sequence logo that we get. So this is saying for our sequence logo, what we've learned is that, in the first position, this part right here is in position 1 of the motif here, position 2, and position 3 here. So these heights, so I have another video on computing these heights, these bits and heights here. But the key idea is that you notice that we do not have any T here. No T made the cut, sadly, because we do not ever have any T here in these sequences. So we're pretty confident that out of these 4, 4 is kind of small, and now we'll be looking at an example with 50 sequences, but we in reality would have way more. But like, you know, in this case, we didn't have any T here because so in this example, we did not have any T here because in the 4 sequences, there was no T given here. So no T at all. And the first, there's a little bit of uncertainty, which is, I hope you can kind of see that uncertainty here, where it could be like either a C here, or it could be a G. You know, C was the optimal one and then G was the runner up. So that's why it's kind of like either like 50-50 in this case, half and half, because 2 of them had a C and 2 of them had a G here. And the height is not as high. Whereas for A in the second position and G in the third position, that's like almost like all, like that's all the time for all 4 out of 4, it's a case. So 4 is a relatively small size. So that's why I'll be showing you a larger example. But all 4 out of 4, in the second position was there an A, so that's like the maximum height. And in the third position, there was a G, so that was the maximum height too. So the key idea that you see is that these heights are the same. But in reality, this height, like this height for the CG for the first position of the motif is smaller than it is for the second and third because there is less information that we know about the first position. We know it could be either a C or a G. It's a lot better than us saying, oh, it could be an A or a T or a G or a C equally likely. I mean, we have tuned it down to 2 out of the 4 bases, either a C or a G. At least we're not doing all 4 DNA bases, but it's still like kind of uncertain between the 2. So it could be between the 2, whereas for the second and the third in this, it's kind of like a rare example where you could have this much accuracy, I guess, that it would be like 100% times a G and 100% of the time it's an A. But here it could be either a C or a G. No, but here it could be either a C or a G. So since there's more uncertainty in this first base, it can be either a C or a G. It's like a coin flip. We don't quite know. There's less information about this. Whereas here we can say definitely it's an A. And here we can say definitely it's a G. And of course, again, this is extreme because it just so happened that all of our examples, I mean, there were 4. So it's not, it wasn't too difficult for this to be all G's and then all A's in the middle. But like we definitely know this. So there's a ton of information that we already know about the second position and a ton of information that we already know about the third position, which is a G here. But for this situation, this could be either a C or a G. Like twice it was a C and twice it was a G. So we have more uncertainty about this. So there is less information that we know. So in a way, the bits are kind of like, how much information do we know? So the more uncertain you are, the smaller this height will be. So if tomorrow we were to, and we'll see this, if we're splitting it among more bases, like maybe a T, then this height would be shrunk down even more, for instance, because there's more uncertainty. But now that it's 2, it's a lot higher because at least it could be either of 2 and not all 4 bases. So we'll see some other examples to hopefully kind of show that more, but the higher this is, the more information we know, and we know more information, the fewer majority bases there are. So the likelier it is that we have 1 base and maybe a second base, then the taller this will be because we know more information. And an analogy is that like, so if it's winter right now, then it could be either snowing, it could be raining, or it can be sunny outside, for instance. So I could say hypothetically 30% chance of raining, 30% chance of snowing, and 40% chance of sunny. But then the key thing is that if it's in the summer, so, you know, again, it could be 30, 30, 30, for instance. But if it's in the summer, we have more information. Like let's say there's just a 3% chance of snowing in the summer. So 3% chance, just to make it even, let me just say, like not even, to make it easier, let me just say 5% chance of it snowing in the summer, you know, just let's give it some value. 5% chance of snowing in the summer, then 45% chance of it being rainy, and a 50% chance of it being sunny. So we've narrowed it from 3 options in the winter down to 2 main options. So we have a lot more information available in the summer. So the height would be much higher for sunny and for rainy in the summer and overall than it would be for sunny, rainy, and snowy in the winter. And the snowy in the summer would be so small, you might not even see it in the sequence logo because it's like virtually very difficult for it to snow in the summer. I hope that you saw how we generated the sequence logo here. From our set of input sequences, we just put those in here. In the example that we were looking at for the expectation maximization, when we put it into the WebLogo, we were looking at the default, not the classic mode. So when we've input CAG, GAG, GAG, and CAG, what we wanted, according to our idea, is to have it as a PDF. Then what we want to do is have a large logo size. And then for the color scheme, the default was auto, but auto, as we saw, looks like this. Not so great. C and the G's are colored the same. So what we would want is we would want to go with classic. That's what we would want to do, classic. So these are the arguments. So these are the motif occurrences that we have, occurrences of this motif. So this is a better way of seeing. So C is in blue, G is yellow, A is in green, and G again is orange, and T would have been in red. So we can see how these, again, it's the same thing, it's just a color scheme that looks a little bit better. So it depends on what you're looking for, but this is the classic scheme. And again, we can see that this is not as high as this, or A, because we're not as confident about the first base. The first base can be either A or G, and there's that level of uncertainty here that we don't know. It could be either C or G, like half the time it's C, half the time it's G. Whereas in this second base, for the 4 4 sequences that we were looking at, it was always the case that A was in the second position, and always the case that G was in the second position. So this is pretty much how perfection looks, height of 2 here. So basically what I hope that you see here is that, you know, when we're putting these in, these are the subsequences, these are the motif occurrences, and that's my little logo there. When you're creating it, we just input it. We can also upload a text file if we want as well. But this is the CAG, GAG, GAG, CAG, the subsequences, the occurrences of the motif that we're looking at. And I selected from the default output as PDF, logo size large, and then color scheme as classic. And then when I did that, I got this, you know, and this is much better in my opinion. So this is how we do it. I was showing you just how the default looks, but, you know, it's better to use a classic scheme. That's ideal, as we were wanting, you know, color scheme classic, logo size large, and output format as a PDF. One thing, guys, that I wanted to really quickly clarify is I know that I say PWM is probability weighted matrix, for instance. But one of the things I hope you guys can understand is that I'm really trying to say position weighted matrix. I'm sorry, I've been kind of interchanging the two. So really PWM, you know, probability weighted matrix, we can kind of consider it like having the background in the non-motif positions here. Like, you know, these are positions that are not motifs. But in the final matrix that we're using to generate the sequence logos, what we would really be wanting to look at would be that final position weighted matrix. So everything that does not include the background. We just want to use the parts that are estimating the motif. And that's what we end up doing. But I just wanted to kind of tell you that PWM really is position weighted matrix. But I've kind of been mentioning it a lot right now, that it's probability weighted matrix. But I've been kind of mentioning it a lot now that it's probability weighted matrix. So I just hope that, you know, you can keep that in mind, you know, going forward, that it's actually position weighted matrix. But if you call it probability weighted matrix, I'm sure that that should be okay too. But, you know, I just wanted to clarify. Also, we see SpongeBob and Patrick, and hopefully you understand how to do this and how we went through in the CAG and GAG and GAG and CAG, the occurrences. Before I go into my second example with the larger sequence, I want to motivate to you kind of why this could be important. And I'll be talking about a very key transcription factor or a key gene that encodes a transcription factor. So kind of the same thing. Transcription factors are like proteins that remain in the nucleus and help with the regulation of other genes as well. And they bind to the sequence-specific transcription factor binding sites. We're talking a lot about transcription factor binding sites. That's actually one of the use cases for this meme for finding these motifs, because transcription factors could bind to those binding sites that are sequence-specific motifs. We can have SPI1, it's a key gene that encodes transcription factor PU.1 in humans. So basically you can call it the SPI1 transcription factor, or PUI1 is the more formal name, but I'm going to just call it SPI1 here. So in terms of gene regulation, transcription factors, these proteins, and they remain in the nucleus of the cell and help regulate the expression of genes, either the activation or the repression. So these transcription factors, they bind to sequence-specific transcription factor binding sites near the enhancer or the promoter of a target gene. So these TF binding sites are often located on enhancer or promoter regions. These transcription factors here coming near the enhancer or promoter binding to the transcription factor binding sites there, they help recruit RNA polymerase II, which then helps transcribe this gene into a copy of messenger RNA that then can leave the nucleus and then be translated by the ribosome into a polypeptide chain that can ultimately, form its own protein or be part of a protein. And I talk more about that elsewhere, but what I want you to see is this is an example of a transcription factor, a TF here, that can bind to a sequence-specific, it has a sequence-specific motif over here, and it binds to its transcription factor binding site here to this motif here, T with A with T, C with G, G with C, T with A with T, C with G. It binds here, and in doing so, it helps regulate the expression of this target gene. That's what it does by binding here, like two puzzle pieces, just like this piece and this piece joined together, just like this. That's what it's kind of doing. This transcription factor, PU.1, it activates gene expression in myeloid and B lymphoid development, which are immune related here. And so yeah, again, this is what this transcription factor is doing here. So it's helping activate gene expression in myeloid and B lymphoids, these immune cells. SPI1, if you look at UniProKB, and I have other videos talking more about this, binds to this PU box, a purine-rich DNA sequence, 5' to 3', so DNA is read in the 5' to 3' direction, GAGGAA, that can act as a lymphoid-specific enhancer. This protein is a transcriptional activator that may be specifically involved in the differentiation or activation of macrophages or B cells. Those are immune cells, and it also binds RNA and may modulate pre-mRNA splicing by similarity. When we have the messenger RNA, that we get from transcribing the gene to pre-messenger RNA, then there's some processing that's done. A poly A tail, 5' untranslated region, exons spliced together, introns, removed. Since this is a purine-rich DNA sequence, I'm just zoomed in on this text here. So this transcription factor binding site, TFBS, is an abbreviation for SPI1. So SPI1 binds to this purine-rich DNA sequence that can act as a lymphoid-specific enhancer. This is this part here. And we call it purine-rich because purines is like pure as gold, adenine and guanine. And you notice here that there's a lot of guanine and adenine here, like GAGGAA, like it binds here a lot. CGAT together, these base pair together, but C and T are the pyrimidines and G and A are the purines. I like to look at George Clooney. This tells you the pairings. And think of pyramids in Turks and Caicos. So T and C are like pyrimidines. So let's see what those look like. Oh, this is beautiful. I want to go swimming there, Yangtze. Let's go. And we're at the beach technically, but it's not as nice as Turks and Caicos. It's a virtual beach. Psi, T and C are pyrimidines, and A and G are purines. So all gold is pure. A and G are purines. And lymphoid-specific enhancers, so again, like lymphoids, they're often like B lymphocytes, B cells, T lymphocytes, T cells, natural killer cells as well. These are immune-related, so it's an enhancer. So these are types of white blood cells that are part of the immune system. So as we mentioned here, SPI1 binds to this purine-rich, it's purine-rich because it's composed of A's and G's here, this TF binding site, 5' to 3'. So it binds to this sequence here. So where it binds is GAGGAA, 5' to 3'. So I kind of looked up an image of SPI1. It roughly looks like this, kind of. This would be SPI1. It's able to then bind to this GAGGAA, to this TF binding site, this motif for SPI1. When it binds here to that, it helps regulate the expression of this target gene. So because it binds here, then SPI1 would have to have some site that could be like
a C binds with the G. This would be a T here. A C binds with the G, C binds with the G, T, and then the T here. It would need something like this.
So basically SPI1 has this PF binding site, this motif, GAGGAA. This is what it's kind of likely that it is. It binds to the sequence, a pretty highly likely sequence though, as a transcription factor. So we can call it SPI1, or we can also call it PU.1. It is super huge, like in Alzheimer's and other diseases. It's a really huge transcription factor. It's involved in microglia, which are the immune cells of the brain. They're the resident macrophages of the brain as well. So basically what we see here is that SPI1, or PU.1, I just call it SPI1. It's kind of equivalent because the SPI1 gene encodes this transcription factor. But the key idea is to see that, as a transcription factor, we can basically see that, it binds to DNA at a TF binding site, which is often what this is, GAGGAA from 5' to 3', and helps regulate the expression of various genes here. And, for instance, this, and then it's going to lead to proliferation, cell death, migration, invasion, all things that, immune cells are kind of doing. So this is a finding here. This is a figure that I generated. And what you can see here is that in chromosome 6, in this position around, this is the base coordinates that we're looking at, around 3837510. In this region of chromosome 6, for instance, we can see the sequence logo that was generated. And this is the SPI1 motif. This GAGGAA. It may not be exactly like that per se, but it's very similar. So this could be GAGGAA. You see, for instance, GAGGAA. And this is where this motif, this is where this SPI1, it binds to this region along the DNA. Now, I was using this for other reasons as well, but it is very interesting to note that, Yangshi, that, these are examples of how these motifs are binding, of how the transcription factors bind to transcription factor binding sites, sequence-specific motifs that they recognize. They have those regions that they can recognize here. And I want you to see that, this SPI1, it recognizes this region in general. And we were looking at examples where it's kind of like, you can see when there's uncertainty, in our case, like C and G and A and G in that example, here there was uncertainty, so the height's not so high, but when it's like definite, the height, shoots up here. But this is the same idea. This WebLogo is sort of something that's generated for this motif, height between 0 and 2. And if the height, this is the top of the height is going to here. But basically, if the height is 2, that means that we have perfect information. If the bits is 2, we have perfect information, and it's usually like just one gene. One nucleotide base. And we have perfect information, based on our 4 sequences that they were either CAG or GAG. So they were always A in the second position and always G in the last position for those 4. And again, it's a very simple example. And in reality, it may be messy. There may be various bases in these second and third positions. So it may not always be A or always be G, but it would be pretty neat if that was the case. But in general, looking at all the binding information that Homo sapiens encode motifs SPI1 known for from this database, they found in chromosome 6 at this location along the chromosome 3837510. These are the sequence logos representing the motif. And these all come from occurrences. And we can see that this A is the highest. Between this and this has more height. So this means that we are more certain, this height is here versus this height. And you can also see that the T is bigger here. So that means there's still a bigger probability of T being in this position than in this position. But there's more uncertainty about the A, even though it's pretty high. This could, for instance, be like 80% chance of A and like 20% chance of T. And this could be like 90% chance of A and 10% chance of T, for instance, and the other bases may not appear. So this is likelier to be an A.
90% A or 10%.
This one, on the other hand, could be more uncertainty here. So there's more uncertainty here about, A. Even though it's a slight difference, this height is taller because we are more confident about a majority base A here. And it's really high, close to 2, because we're kind of singled it to A or T, but T is so small. Here, C and G are very small probabilities. They probably are like 1% are very small, so they don't even appear here. But, we are more confident in this position, more information than in this position, because the chance of having the other base like T is slightly higher here. So there's less certainty about A. That's why there's a little bit less information. It could be like 75% A. Probability of having an A in that position could be 75%, and then 25% or even 20%. But let's just pretend there's these two bases and the other ones are so small. The probability weighted matrix, it could be anything.
The probability weighted matrix is just like
you have these probabilities in a given position and you want them to sum to 100%. This was a motif, for instance, then this would be, summing, this would be like sum of these values here, which would be 100% roughly, maybe slightly off for rounding, 100%, 100% if you add these up here. Then 100.10 though. So basically what we're seeing here is that, we just are looking in a given position, and if there's more certainty or less certainty, and we'll look at this example, that's how we expect these sequence logos to be based on. So if we're more certain about a given base, we expect it to be the kind of higher. So here, for instance, the height is less because, 75% chance for A, I'm just guessing in this case, 25% chance of T, and let's just say 0 for C and 0 for G. So we're less confident about A. I mean, A is still majority, but we're less confident about it because it's 75%, but still a 25% chance of T. So this is much higher here, but the amount of information we know is a lot less. 90% chance of A being in this position, maybe 10% chance of T. Then we see here as we go down, this is decreasing overall. This behavior here is decreasing. Here, the percent chance of A is decreasing and the percent of T is increasing here. That's why this height overall is decreasing. So that's a way to see it. But in general, the SPI1 motif here, it's pretty likely to bind to GAGGAA. So GAGGAA in particular is what it usually found to bind to, GAGGAA, for instance. That's what would maintain the integrity of it binding, GAGGAA, for instance. But it's in general, it usually binds to purines. It could bind to a pyrimidine like T here, but this is just an example of a data resource, and it shows like a sequence logo, and SPI1 motif could be this here, and SPI1 could then bind to that. The things are not always black and white. They could be shades of gray. So this looks like the motif for SPI1. But the general idea here is that, this is what we typically assume is the, binding site motif for SPI1. In general, it is composed of a lot of, purines A and G. And I wanted to show this to you too. So this is from an interesting paper here, and I've shared the link here. But basically in this paper, they're looking at like, SPI1, for instance, again, and they're saying the SPI1 and RNA polymerase 2 exhibit preferential binding in the genome. And then they're defining the definition of each genomic region here: enhancer, promoter, infragenic, 5 kilobases downstream as well, transcription start site, for the gene as well. And the key idea is that what they're saying here is that we want to focus here on this example. I wanted to just give everything else in context in case you're interested, but we're focusing on this part here, on E here. And what I want you to take from this is the following. In E, we see the sequence logo bits. This is 5' to 3' here. And what we see here is they're saying sequence logos of the probability weighted matrix produced by this chipmunk software using all 17,781 SPI1 peak sequences. They generated the sequence motif here for SPI1. They used 17,781 peak sequences. And then they generated this WebLogo. So we just put in 4, in our example, we just put in 4, CAG, GAG, GAG, CAG. But here they're putting in like 17,781 peak sequences, motif occurrences, where they think that SPI1 activity is the strongest. And they put that in here. And then this is how they're figuring out GAGGAA, GAGGAA. It is along here. So we can see that along the motif, what they have gotten is kind of like consistent in a way, like, this GAGGAA that they talk about, it does fall along this sequence logo, GAGGAA. This probability weighted matrix threshold corresponds to a false discovery rate of 10%. So we saw this GAGGAA. Again, that's what they consider like very often to be the transcription factor binding site for SPI1. So I just wanted you to see that here. So next, I want to talk about 2 more examples. One is an example of running a larger scale, running this multiple, this expectation maximization for motif discovery running meme. And the last one will be on the Gibbs sampling example as well. And the last one will be on the, so the next example will be on a larger scale, expectation maximization using meme. And the last one will be using Gibbs sampling. And these are examples that, I hope that you can go through. And if you have any questions, just please let me know as well. But, they're based on videos where I've gone through and I've talked about how we get the probability weighted matrix using the meme algorithm, or kind of like a newer iteration of it, which is, the Gibbs sampling approach. But I wanted to show you because we talk here about like so many sequences. So we had just used 4, but people are using a lot of peak sequences for a transcription factor to generate like a sequence logo, which again has like this GAGGAA as well. So in this example, we are going to now see what happens if we look at 50 input DNA sequences. So no longer will we look at those 4. We were looking at 4 before, 4 sequences, but now we're going to look at 50. So we're trying to make it a little bit more realistic so that now we can see another example of generating a larger sequence logo and then kind of interpreting that. So these are 50 input DNA sequences. So each of these is 20 bases long. So we're looking for a motif of width W equals 6. So this is what we specify. We could have looked for a width, from 1 to 20. Similar to in the other example, our width was actually, our width was 3, but our length was like 6 bases. But like we specify 6 here for our motif width. It could have been any number between 1 and 20, but you have to be kind of like careful with what you choose because that can impact what the model in the end. But anyway, so 50 input DNA sequences, 20 bases long as a length, motif of width W equals 6. So from this information, basically from these 50 sequences, so these are 50 sequences right here. Oh, these are 50 sequences. And we're going to learn from these. So we're going to end up having motif occurrences, for all 50 sequences. We're going to try to use the expectation maximization approach to find the optimal model. So expectation maximization algorithm for motif discovery mean, it resulted in this probability weighted matrix, our final model. And the motif has width 6, so there are 6 bases in the motif model in this probability weighted matrix, this PWM here. This is our PWM. That's what we see here. This is our background, a non-motif position. Anything that's not in the motif. So either it can be in a motif position or it cannot be in the motif. It can be an occurrence of the motif, or if it is, then it would be here. Otherwise, it would just be a background. So background is everything else. So when we go through this with expectation maximization, 50 sequences, motif width of 6, for these 20 bases, then what we got is this corresponding final probability weighted matrix when we ran it to convergence. So again, a probability of A in the background is 25%, C in the background in a non-motif position is 27.6%, for instance. And then these are positions in the motif. So we can kind of get an idea if we were to just kind of look at the highest one. We could say, oh, it looks like, in the first position of the motif, we have a 40.5% chance of a C there. In the second position of the motif, we have a 51.6% chance of an A. So that's the highest here. In the third position of the motif, we have like a 91.9% chance of a T. That is pretty high. And you can see that the others are pretty small, right? Like, 2.2%, sorry, 2.2%, 4%, 2%. So very small. And this is like really high. So you're pretty confident in the third position in the motif, it would be like a T, pretty confident, like, pretty high chance that that's the case. But even higher is in the fourth position of the motif being a C. Everything else is about like 2%, roughly 2%. But in the fourth position of the motif, and again, the way that you represent this probability would be like, this would be a probability of C, probability of having a C, a giant C, not the character C, in the fourth position of the motif. And then, for instance, this would be the probability of having a T in the third position of the motif, for instance. So similarly, we go through and we see the highest one. So here, this is also pretty similar. It looks like C and C again here. And lastly, well, this is not quite too high because, this is 53.1% and runner-up is G. So probability of a G in the last position is, in the sixth and last position is 53.10%. But like a T is a runner-up, at 30.6% probability. So it's not quite as high. We would expect the height to be a little bit lower. So that's kind of like how we would see it here. But based on this model as well, on these 50 input sequences, choosing a width of 6 as well from this motif width, running expectation maximization, and getting this probability weighted matrix, we learned these following positions within these sequences where our motif occurs. So this is saying like in the 11th position in this sequence, that's where the motif starts, and the motif will be 6 bases long. 1, 2, 3, 4, 5, 6. And based on this is like our Z matrix. And then based on this, we learn the subsequences. So we basically, go see, like in this example here, we can see that like, in the third row, we can see in the third row, this is 2 here. So this 2, for instance, it corresponds to this 2 in the third row. It corresponds to starting at position 2, and it is C, T, T, C, C, G. So we have to go here, and then we have to look at it like this. So let me actually show you kind of juxtaposing them all together, actually. That might be better. we have our example 1 here. Okay, so what I want to do is just focus on this, for instance. We want to just, because this is 2, so this is a little bit easier to count. Let me just make this actually red, make it a little bit easier to see. So this is 2 here. And this is the corresponding, what we learned here, the motif position here. So what we want to then say is that, because it can be 0, 1, 2. You just have to make sure this is what it is actually. Because the code was run in Python, so the numbering is 0, 1, 2. So everything is off by 1. But don't worry, you just have to be consistent. So in the end, we extracted this position. Here, that just means 0, 1, 2. We start here. It doesn't really impact it as much. We just need to know what the indexing starts at here. But just don't worry about the technicality. The bottom line is that this motif that we learned is C, T, T, C, C, G here. So, and again, in this example, like if we were to look at like this one here, then we would start like, we need to go at 0. T, 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 C, C, G. That's what it is. Right. And this is T, 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 C, C, G here, which is the third 0, 1, 2, 3, 3, and we go for 6 bases because that's our motif width. So you just have to be consistent of what you're starting with, but that's just kind of like an example there. And so we learn these subsequences. So these are occurrences of the motifs that we learned along with the probability weighted matrix that we got. So basically, we learned, this is our Z matrix of the positions. And then we get these subsequences that we learn. These are the motif occurrences, 50 motif occurrences here. And we're going to generate the sequence logo for this largest set of subsequences. So what I'm going to do is I'm going to go here and I'm going to take these subsequences that I learned. So I'm going to just basically come here and take these subsequences here, these 50, 50 here, and I'm going to put them into the sequence logo generator that we were looking at, that WebLogo. So let's go there. So again, we saw this old motif that we were looking at. Now let's put in these 50 sequences, subsequences. These are the 50 motif occurrences here. And we're going to try to create the web logo. We save it as a PDF here, large logo size, and then classic scheme. So let's create a web logo, Yangshi. Let's see, we're going to create it and see what we get. So this is what we learned. Yangshi, this is what we learned here. I'm actually just going to have it as a PNG, high resolution, so that I can like copy and paste it easily, to copy the image. But basically, what we ended up learning is this sequence logo here. This is pretty nice. It's pretty big. A lot of information here. So this is the sequence logo. I'm gonna just move this up to the probability weighted matrix so that we can kind of see how that looks. And see if the intuition kind of makes sense to us as well. And these are positions along the motif, and then this is the height here, the information that we have. So if we were to go across a number, then we would see that this is like the first position of the motif, second, third, fourth, fifth, and then sixth, the sixth and last. And remember what I was saying. So we had gone through in a way and we kind of identified the maximum. So maximum is here in a way, and then here, but not by too much. Here, 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 and here in a way. Sorry, the color is not so great, but like we can see here that the maximum is a C. Actually, how about I do it like the top two? So the top one. So the top is like, this is the C here. Let me find where else C was high here and here. Then let me also look for where T was high. Its value was high. So T was kind of high in the third position here. I think that's about it. And then for G, it wasn't so strong, but like it was, G was like here, for instance. And then A just had a small relative advantage here for A. You know, again, this is for these positions of, you know, our little guy is happy to learn. 1, 2, 3, 4, 5, and 6 again. So what we can see is that in this position, you know, C was 40.5%, but it's not much higher than the T here. So we see that the height is not as great here. And this example, A, but C still has a relatively slightly taller height than T does. They're really close, just off by like 2.2%. You know, C is 40.5% and then T is 38.3%. So the top one is usually the majority one, the top in the sequence logo. So it's like the main priority. It's like the top of the pyramid, the main guy, right? So this is the 51.6%. A is high, and then followed by T here. T is 44.5. So A is slightly taller than T. Again, it's kind of split, but here the majority base, the most frequent base, its probability is rather higher from 40.5 to 51. That also kind of bumps things up a bit because the majority base, the most likely base that we are predicting, A, has a greater probability of occurring than the majority base of C here. So the most frequent base is even more likely to occur. So this gives us more information. If we had to go with the gut instinct, so the way to think of it is if we had to go with the most likely, with the likeliest base, the highest probability, how certain would we be? We would be less certain with C because it's a 40.5% chance of it being there. But here, with A in the second position of the motif here, we're a little bit more certain because it's a 51.6 probability. So much higher by like 11.6%, by 11.1%. But in these cases, we're like super likely to have a T in this third position. All the other DNA bases are kind of like squashed. You can't even see them. And here you probably don't even see the other ones because it's so likely to have a C in this position and a C in this position, the same pretty much here. And lastly, the majority base here is a G, but it's not too much higher above than the T, which is 30.6%. But you know, this 53.10%, it's more than this 40.5%. So the majority one is kind of higher. So in some way, if you were to see it like this, if you can compare it this way in a way, you can see that this is much taller than this. You know, like this height here is much taller than this height. Also because what you're seeing is that this is 53.10 for the majority base, but this is 40.5%. So it's not so likely. And it does have like a spread of the other 4 bases as well. Whereas this one is kind of split between 2A and T. So whereas here it's like it could be G, or it could be T, or it could be C and A. So the second base is not so likely as the second base here. That's why this is kind of higher than this. It's kind of like weighing how much information do we know? Like here, it could be like kind of like 3 bases, like it could be G, T, or A, kind of, or it reminds me of Grand Theft Auto, a game that my cousin used to like. But here it's kind of like more split between 2. So even though this is less than this, it's like, you know, it can be between 2. Like it's like summer, you know, it can be like rainy or sunny. Probably not like snowing. But in this case, it could be like rainy, sunny, or snowing or something else, right? So it's a little bit more uncertainty here. That's why this height is lower. So that's like a key thing to note. So that's how we generated the sequence logo again for this. Just put those subsequences inside here. And then we generated them. And this shows you again some uncertainty. And Yangshi, if you can do this, drum roll, please. Woo, woo, yeah. On to the last example of, kind of finding a sequence logo here. And this would be like a Gibbs sampling approach. I have a lot of videos talking about this and solving this by hand as well. So I hope that those will help you as well. So I think we may have gone through this example in another video, which I'll be showing you here. But this one is like medium scale. So it's like the Goldilocks example. And the key idea in Goldilocks, Yangshi. So Yangshi, a key idea in Goldilocks is always, sometimes it's better, little guy. Sometimes it's better to have the middle, not always an extreme, like not always like too many sequences or too little sequences, but let's have a middle. We looked at 4 before, then we looked at 50 right now. But now for Gibbs, let's make it the middle. So both 4 and the 50 examples, they were for me, but the multiple motif elicitation, using expectation maximization. So now let's just look at Gibbs sampling for a middle number like 10. Let's see that. So it's not as small as 4, but not as big as 50. And definitely not as big as like 17,000 that was used in that paper. So let's look at this Goldilocks now. So Gibbs sampling for motif discovery. And this is a direct problem that you guys have looked at too. So, this is the Goldilocks, having the middle neither too big nor too small, just right. So in this Gibbs sampling problem, please suppose that you're running the Gibbs sampling algorithm for motif finding on a set of n equals 10 input sequences with a motif width, W, of 4 nucleotides. 4. In the A, T, G, C are the nucleotides, but here the width is 4. Further, please suppose that the current state of the Gibbs sampler is as shown below, with the motif occurrences indicated by these red underlined nucleotides. With the motif occurrences indicated by these red underlined nucleotides. So these are the sequences 1 to 10. These are the motifs as of now, and the rest are background, as I was telling you, and I go more in depth when I talk about the Gibbs sampling, but these rest ones are like background, so we can kind of ignore them right now. Because in the sequence logo, we're only looking at the motif parts, not the background. Again, these motif parts kind of get the spotlight, but we're not looking at these yellow parts. Yellow are the background. We are only concerned basically with these motif occurrences that we're seeing, these. So, and we're looking at this in Gibbs, and you might recognize these sequences here because we've looked at this in some other practice problems. So please suppose that in the next iteration of the sampler, sequence 5 is picked, and the motif position in that sequence is to be resampled. So we're going to be resampling this. So these are the 10 sequences, and in red are these motif occurrences as of now. Like if we had to freeze it in this current state of this Gibbs sampler, which is another approach for finding probability weighted matrices as well. So this Gibbs sampler is another approach. So actually in this article that I was talking about, they talk about how, one popular implementation of expectation maximization mean performs a single iteration for each n-mer in the target sequences, selects the best motif from this set, and then iterates only that one to convergence, avoiding local maxima. This partially enumerative nature of mean provides some assurance that the algorithm is unlikely to get stuck in a poor local maximum. Additional motifs present in the set of target sequences can be found by masking the sequences matched by the first motif and rerunning the algorithm. But instead, the Gibbs sampling is a stochastic implementation of expectation maximization. So Expectation maximization takes a weighted average across all subsequences, weighted with the current estimate of the probability that they belong to the motif. Gibbs sampling takes a weighted sample from these subsequences. So we look at it here as well. And there are various things as well that we discuss in other videos, but I just want you to see that these are different ways for kind of estimating, motif models from a set of DNA sequences, a certain motif width. So right now, if we had to freeze it, we're in this next iteration of the Gibbs sampler, we're picking out sequence 5 and the motif position in the sequence 5 will be resampled. So we're going to be using, in this case, a pseudo count of 1 for our estimate. So we add 1 just to like fluff up all the probability calculations so that we don't have any 0 probabilities for any basis. And we assumed a standard probability weighted matrix model, so no prior information was used. I do talk in other videos about palindromes and standard probability weighted matrices. So Yangshi is saying, please check those out. Please subscribe and check those out as well. Those will really help you guys, hopefully. So here, cute little guy. What we're doing is we're going to compute these parameter estimates. Basically, if, we're doing this predictive update step, given that sequence 5 has been selected. So I'll talk about how to do this. Essentially, we eliminate sequence 5. We just get rid of it because we're holding it out. So we're looking at sequences 1, 2, 3, 4, 6, 7, 8, 9, 10. And then what we're doing then is we're just focusing on these sequences here. And in particular, only the motif parts right now, because we want those to be our subsequences, our motif occurrences as of now. So again, I do talk in other videos about how we derive this probability weighted matrix here that I'm going to show you, and I talk about how we can hold out sequence 5, in this iteration of Gibbs sampling for these same bases. So please subscribe, it's free, for more updates. And again, we ended up talking through how we get this probability weighted matrix and the formulas that we use. So we have the background and the motif, and we can also use these motif occurrences here for these 9 sequences because we end up using these 9 minus sequence 5 to get this probability weighted matrix. So I thought it would be interesting to show you what the sequence logo looks like for these 9 sequences. We're not looking at sequence 5. I show you how essentially we just kind of like cut it out and we don't even really consider it here. It's like when you're holding out a sequence, you're not using it for the estimates. So including all of these together would be kind of a wrong approach because we're not even looking at 5 here. We're deriving the probability weighted matrix, essentially not using that here. So what I then end up doing is I just get this I get these subsequences here, from these are the motifs. And we can just kind of throw this into, again, the sequence logo and see what we end up getting here. So let's do that as well. And let's see if we understand kind of what we ended up getting. So we can just go back here, we can clear it and we can paste it here. We come to this link here. You ideally, if you want to save it as a PDF, that's the highest, but for now, just to copy it, I'm going to just make it a PNG. We want large logo sizes and auto scaling, and then color, it should be classic. So we create the web logo here. And this is our web logo that we learned. This is our sequence logo. It's pretty neat, right? So for our sequence logo, let me paste it here that we generated. I'm going to just paste it here. So again, for this sequence logo, we do not really look at this background probability. We do not look at this at all. We're just looking at the motif part. So we're looking at this part here. K equals 1. This is 2. And then this is 4. 1, 2, 3, and 4 here. Let's see, it's kind of higher here, but not by too much. Then, in the second position of the motif, A is actually higher, so it's going to be stacked at the top. Then again, G is now going to be taller for the third position of the motif here. And in the fourth position of the motif, it seems like it's kind of a tie between C and T. So it's probably just going to choose one of them, which it does C here. Like, you know that if something's at the top, it would be either the most frequently occurring or kind of tied with the next base. You can kind of see by the height here. So C and T are both tied here. So C is 30.77% chance of C being in the fourth and last position of the motif. And T is also 30.77% chance of being in the fourth and last position of the motif. So they're the same. So they have like the same height here. And then followed closely by G in the third position, and then A small. But like, you know, if something's at the top, it can either be like the most frequently occurring, as it is the case here, or it can be tied with the next base. The second base, it would not be the maximum. If it's in the second position, it's either in the second spot or it's tied with the first, for instance. Like that's how you can interpret it. So if a base is at the top, it is either the highest probability or it's tied for the highest. In this case, it's tied. So C is here. But since there's so much uncertainty, like, you know, we could be either C or T up pretty high, and then G is coming behind it. The amount of knowledge and our confidence about this is the lowest, actually. We don't really have a high confident one. It's like we can either say C or T. If somebody asks us to choose, we would be like, let's flip a coin. So we kind of have very little information here. We just kind of can tell them that probably don't choose A, maybe not G, but it can be either or. Whereas in the third position, we have higher knowledge, like a 61.54% chance of having a G in this position. And again, all of these sum up to 100%. If you sum up the rows, it's basically in a particular position, what's the probability of observing a given base? So this is 61.54%, which is pretty high for this majority base of G. And then an A is 46.15%, closely followed by T, on a kind of like half, you know, 23.08%. So it's not as high here. And you can kind of see something similar here where in a way, let me just color them kind of yellow for now. So you can kind of see, you can see that this here and this here right now, that this and this tend to be the same. So this is a C here, and this is an A here. And the next base is a G, which is second highest, and then this one is a T. So it's like roughly similar, but it's just like shuffling of which bases. So 46.15, 46.15, 23.08, 23.08, 15.38, 15.38. So it's the same. So this is the same exact height. It's just that the bases are differing here based on what we are selecting. But this is essentially a sequence logo. And I just want you to understand that this is how sequence logos are. And different transcription factors, for instance, they have their own set of sequence logos. So sequence motifs are short, recurring patterns in DNA that are presumed to have a biological function. Often they indicate sequence-specific binding sites for proteins such as nucleases and transcription factors. This is a key focus. I'm really interested in this too. So we can see, for instance, if we look at transcription factors, these are the sequence logos, and now you can better understand how we generated them. You can see like this is NFIL3, FOXF2, FOXB1. This is interferon 2, for instance. These are sequence logos for various transcription factors. There's very little information here, but there's a lot of information about these bases here. The higher the height, the more information there is. And often we take the one at the top. So TTA, TGTAA, for instance.
Other examples of transcription factor binding sites for various transcription factors like NRSF. They look at NSRF binding motif with 17 base pair spacer between half sites. So these would be 17 base pairs in between. And then here instead it's an 11 base pair spacer between half sites. This is the binding motif for EWSFLI1, you know, this transcription factor complex. And they see that it's a GGAA and tandem repeat. So what this is saying is that we're repeating GGAA again and again, multiple times. GGAA, you can see that we have all these blocks of GGAA. In this case, this would be 1, 2, 3, 4, 5, 6. So this would be GGAA. And this is like a canonical ETS family motif as well. You can see it's kind of a little bit different. So the motifs, so the motifs are kind of different, but the sequence logos give us this type of information that's helpful. And we can see like the binding consensus sequence for PAC6 as well. You can see like there's a Tata box and PAC6 is binding, and people are trying to figure out the consensus sequence here. And you know, this is kind of showing conservation plots, just data analysis here. We can see the site name, sequence, matches, e-box, binding consensus sequences. So sequence logos are everywhere. This one is also kind of showing how we can map DNA sequence to transcription factor binding site, binding energy in vivo here. So regulatory context can alter sequence preference. So they also look at the sequence logos for two binding sites, indicate nearly identical sequence preferences. So we can see like this XYLR here, what it's binding to, you know, on the DNA, RNAP on the left and on the right. We can see these sequence logos are just used to represent like LAC1, upstream, downstream. What are these binding motifs looking like? If it binds upstream, it looks more like this. If it's binding downstream, it looks more like this. And for this PURR here, for instance, if it's binding mid-RNAP, for instance, I know like I'm just kind of showing you this example, but I'm just trying to show that if something binds, for instance, like mid-RNAP, this is a sequence logo here, but if it binds downstream, this is what it looks here. So the idea is a regulatory context can alter the sequence preferences. So different transcription factors, or even the same, so different transcription factors bind to different sequence logos, but even the same transcription factor could potentially, you know, like per or LAC1 here, you know, LAC1 is this purple one here, per R is this orange guy here, XYLR here, and here, the same like factor, the same protein can bind to different regulatory sequences, to different binding sites based on the context, based on an XYLR. If it is left, it's a different context than if it's right. You see like these sequence logos are different. If it's a per RR, if it's binding mid-RNAP, for instance, if this guy is binding here versus binding downstream, the sequence logo is different. What its preferences are for binding is different. And for instance, for this LAC1, if it's binding upstream, this is its sequence logo. But if it's binding, this purple guy is binding downstream, then this is its sequence logo. Based on where it's binding sites can differ, and that's key. In general, people can ask, how do you find the motif of a DNA sequence? So we can use established lists of motifs to search our DNA sequence to see what we can already find. But if people had to start out with them, we can also just discover them from scratch using algorithms like Gibbs sampling and expectation maximization, which we talk about in other videos. So we can derive a consensus sequence or probability matrix. And others are showing about like how motifs can show high functional specificity.
I hope that this video was helpful for you. The key idea is that you take the motif occurrences that you have, you come here and you just throw them in here on this website, and you can get the link that you're interested in. You just have to put in the occurrences of the motif. It'll figure out like how many bases, like 4 bases in this motif. It only has to be for those motifs, those subsequences. You put this in here, and then you can specify different colors. Like if we chose like auto, like in the beginning, it's just looking like this. If you look at different color schemes, we can even see chemistry, amino acids. It looks like that. And chemical breakdown, base pairing, maybe CT are pairing together and AT are pairing together. So that's a base pairing one. So I hope that this is helpful. So I have a lot of other videos on these topics. So if it was helpful, please like, please subscribe. And again, please reach out with any and all questions. Even Yankee says so too. So thank you guys. So thank you guys for watching. So yeeks. So thank you guys. Thank you guys for watching. There's a hug from cute little Yankee. He's so cute. Thank you guys. And hopefully then there's a heart, and you see from him, his little sign off logo is these peekaboo, peekaboo, and peekaboo. Thank you guys. I'm Saniya, and again, this is cute little Yankee.